Hello, everyone, and welcome back to ImmunoLogic, the podcast where we take the cutting edge of immunotherapy and try to wire it into real-world clinical insight. That's why my delightful co-host, Janna, is here. I'm Joe Fraietta, your immuno-obsessed host who is always looking for the next way to supercharge T cells to save the day. You know that's my favorite topic. So, we often talk about in this field of T cell therapy, boosting the immune system, right? Just very generally. But, usually that's like, I see it as firing off a flare gun, right? And rallying every immune cell in earshot, if you will, right? But what if we could be a bit more exclusive? So imagine giving your cancer fighting T cells, I don't know, a private communication line, secret growth signal of sorts that only they could hear and respond to. Right? So it's kind of like having a custom high octane fuel that only your race car can use, leaving all the others on the track unaffected, right? So that's the idea of personalized fuel for T cells. And that is actually the heart of today's episode. So before we dive in, as always, let me introduce my partner in crime, the ever insightful Dr. Janna Minehart, in the clinic by day and in the lab by night. I don't know how you do it. It is such an absolute pleasure to welcome, can I say old friends, current friend, but we were in different labs right next to each other in the open space concept. So it's my my privilege really, again, to welcome Dr. Anusha Kalbasi. And he's an associate professor of radiation oncology at Stanford University and a physician scientist, if I may say, at the forefront of innovative T cell therapies. It's really been fun to follow your career. And so over that time, I feel the way that I would describe you, Anusha, is that you've really blended and bridged the worlds of oncology and immunology all the way back from when you were studying how radiation, right, can modulate tumor immune responses, to then going on to do some like amazing work, really pioneering genetic strategies that reprogram T cells for better persistence and potency, and that's what we need with these living self-replicating drugs. So a little bit more. So in 2022, Anusha co-led a landmark study that introduced the synthetic interleukin-9 receptor system as a way to supercharge CAR T cells, right, without the usual need for the chemotherapeutic conditioning that we've talked about on previous episodes. So now, actually, I must say, Anusha is the senior author of a new study just out in Immunity, I think a few hours ago, right, Anusha?
That's right.
So it's really fresh data here. But this paper, and I read the preprint, takes the concept even further. So using IL-9 really is a naturally orthogonal cytokine to selectively expand engineered T cells. And again, the goal would be to eliminate the need for the lymphodepleting chemo and, of course, greatly enhancing anti-tumor efficacy. So in short, Anusha, I feel like you've developed a whole new playbook for making CAR T cells work in solid tumors by giving them their own private growth factors. It's really brilliant. And so we are thrilled to have you on the show. Welcome to ImmunoLogic.
Thank you, Joe. Thank you, Janna, for having me. This is a lot of fun. It's, being at Penn those years ago was a huge inspiration, being that ecosystem and your presence on that floor was definitely something I'll always remember. I mean, you were the quintessential member of that floor, if I recall.
Uh-oh.
I bet you could hear his giggles.
Giggles among other things, but
yeah. It was a stressful time. Fellowship, postdoctoral fellowship is a stressful time, but somehow we got there. I don't know where I got there, I should say.
So it's fun. It's fun to come back full circle.
Yeah, definitely. So, can I kick us off, Jen, or would you like to kick us off? Up to you.
Go for it.
Okay, so let's start with the big picture. CAR T cell therapy has been, as we know, a game changer for blood cancers. It's trickier than solid tumors, although, as I've said before on the show, we're really starting to turn the corner. So one major roadblock is that when we infuse T cells into patients, they often won't expand. So that's one, hurdle. That's where everybody's doing their CRISPR screens in the expansion phase. Story for another day. But really persistence, right? They often don't persist well unless we first give patients a hefty round of lymphodepleting chemotherapy. It's often cyclophosphamide and fludarabine. And the idea is you're clearing out the patient's own lymphocytes to make space and provide homeostatic cytokines, these signals for incoming T cells. So it works, but it's toxic. So this is why I'm so interested in this concept of an orthogonal cytokine signal. Can you unpack that a bit for us?
Yeah, no, I mean that was our original motivation back before, we even got into IL-9 was actually there had been labs that had discovered these synthetic ways to have cytokines talk to receptors, and the idea was actually to, get around the issue of toxicity because we have a ton of cytokines that we can play with, and I think of it as kind of a spice rack. You're in your kitchen and you have all these spices that you can use. Unfortunately, for most of these cytokines, when you give them to patients, they cause a lot of toxicity. So it's a hard road to go down clinically. And so there were several brilliant engineers, one of them being Chris Garcia's lab, who developed kind of this orthogonal system where you have a synthetic cytokine that has a synthetic receptor. And then if you engineer your cell, then you can talk, like you said, the perfect way of putting a private line to your cells. And when you can do that, you get around the issue of toxicity, which was great. But when we saw that, we thought immediately, oh, well, maybe that's a way to get around the idea of lymphodepletion, because the whole point of lymphodepletion is to get rid of all the other cells that cytokines talk to so that cytokines can only talk to your engineered T cells. And
Yeah.
so that was the initial motivation. But actually, our first finding there was kind of surprising in that just talking to your cytokine or talking to your cell with a cytokine is not enough. You need to have the right signal. You need to have the right flavor. And it's not just about getting your cells to grow and expand, but it's about getting your cells to do the things you want them to do, get into tissues, get into tumors, see antigen, be able to counteract, all the other hostile issues that cells encounter in the tumor microenvironment and beyond. So the question really became, okay, what is the right spice, right cytokine signal that we can give to
Yeah,
T
why
cells?
IL-9?
Yeah. And that was something that we kind of stumbled on because we tested a bunch of cytokines in the same family, the common gamma chain family, and we stumbled on the IL-9 being just really potent, and it was kind of surprising to everybody because nobody had heard of IL-9 really,
Right.
at least in our kind of spaces. People obviously there are people who've dedicated their careers to IL-9. It's been involved in allergy and asthma and fungal infections and other things, but generally not in T cell therapy. So we were excited about that. But when we published the paper, it was funny because the immediate question was, well, we're using this complicated synthetic system to deliver IL-9. Do we really need that? Can't we just use regular IL-9? And so
Mm,
that's kind of how we got to where we are now.
yeah. Because I guess for instance, IL-9, right, in some studies has been noted to recruit certain myeloid cells, right, or mast cells.
Mast cells,
I
yeah,
think
mast
so.
cells.
Yeah. Um, so regular IL-9 can specifically feed the, well, not specifically, but why don't you get toxicity, I guess is what I'm asking in a bad
Yeah,
way.
so we that was the first thing we did was like, okay, let's try to give IL-9 to T cells and see what happens, and nothing happens. And then we started looking like what cells actually have the receptor for IL-9. And we looked everywhere and there really weren't any cells that had it at high levels. Even the mast cells and memory
Ah,
B cells that people had reported, it was in like a minor subset of the cells. And then we looked at various data sets and it was kind of interesting. It almost seemed like IL-9 was this evolutionary afterthought, like it kind of had
yeah.
just been discarded from the system.
The
And
appendix of the
so
immune system.
yeah, exactly.
That's a good way to put it.
And I'm probably angering some hardcore IL-9 people that work on asthma and allergy and the role of IL-9 in those diseases. But for our purposes, it seemed pretty kind of dispensable. And so we thought, well, let's just engineer our T cells with the IL-9 receptor and use IL-9 in the same way that we were using that synthetic cytokine receptor pair. But if you have to think about it, when you make synthetic receptor pairs, you have to make them really weak because you need to make them in a way that they won't act on their natural receptors. So they end up being these really attenuated, these really weak signals. But by using the wild type kind of IL-9 and IL-9 receptor, which is actually a really strong pair, you're taking advantage of nature's bounty. And so it turns out that that native cytokine receptor pair gives a much stronger signal, and the cells do, just blow away the competition being the synthetic IL-9 pair. So that was kind of our first, okay, this is something different. This is something real. And then that led to several other kind of things that came from that.
Right.
So to
And
take a
so
step
from your,
back,
oh, go ahead. Go ahead.
to take a step back, you're introducing the kind of the native IL-9 receptor to T cells
Yeah.
who wouldn't otherwise express it, and then using
Right.
native IL-9 to then
Fuel
uh,
them.
fuel them. Yeah.
Yeah, exactly. And it turns out everything we did with the IL-9 in our tumor models, when we just gave IL-9, nothing happened. We could give huge doses of IL-9 to mice and the mice would be just fine, in contrast to some of the other cytokines like IL-2. But the tumors, would grow unless we engineered our CAR T cells or our TCR T cells with the IL-9 receptor and gave IL-9 together. That pair was what, drove really, really potent anti-tumor immune responses. So it was
What's
really
the kind of
that communication line.
Yeah, what's the kind of downstream signaling,
Yeah,
That's
simplistically?
what I was going to ask, right? So what I remember from your prior work is that you activate like a unique trio of stat signaling, right? Like so stat one, stat three, stat five all at once. And that caught my attention because we use typically in the field like IL-2 or IL-15 signaling. That's
yeah,
mostly about stat five, a little bit of stat three, and IL-7 is mainly stat five too. So,
right. Yeah,
IL-9, when I read your beautiful work, again, it seems to be that triple threat, right? You get like stat five
right.
for proliferation, stat three maybe for stemness. That's important. We think it is. And what the stat one possibly for anti-exhaustion or even better effector functions.
Actually, so we should come back to the stat one part because so
Okay,
now this is
got that
now
wrong.
we're getting under the hood. We're really getting into the mechanics of it, and that's the stuff that like we nerd out on all the time. But that exactly that mix was interesting to us. And particularly because for a long time, the field has kind of been obsessed with getting cells to grow and expand and proliferate. And that's like been the holy grail. Get T cells into a patient and get them to expand. And how much did they expand? And that's going to tell you if the patient's going to do well. And what we were surprised by in our initial study was that the IL-9 doesn't actually get the T cells to expand very much at all. It's a weak proliferative signal, but it worked just as well as the really strong proliferative signal. So we think that there's more to it than that, and those other stats probably play a role. And in our first paper, we didn't really get into the mechanics of what stat does what. We just found that, it seemed like T cells had this blend of stemness and effector function, and we also had these multiple stats. So we kind of connected them saying, okay, you have multiple stats and then T cells have multiple functions. The nice thing about our new work is that because the signal is stronger, we actually uncovered a fourth signal, which is STAT4, which we didn't really notice in with the orthogonal synthetic system because it was a weaker system. So in fact, we have STAT1, STAT3, STAT4, and STAT5. And I'll mention one more thing, which is that in addition to being those four, it's really just those four. A lot of cytokines have other kind of bystander pathways that are activated, MAP kinase, AKT,
Right.
ERK. IL-9 is really just the stats, and that's like, one of the canonical cytokine, kind of the classical cytokine pathways. And so that was nice for us. It was like a clean system. It was a really pure system. And then on top of that, there's this like sequence of four amino acids in the receptor that drive all four of those stats. So basically, everything comes down to these like four amino acids. There's really one amino acid, but the amino acids around that one amino acid can kind of dictate the balance of the stats. So it's
Right.
like this
What's the
perfect
advantage of that?
system. Well, we can then, I mean, as you know, we have so many tools to edit genes now.
Hmm.
So you can make manipulations of the sequence right around that key amino acid and change the flavor of the stat 1, 3, 4, 5 mix. And
Yeah.
that provides us a really great tool to be able to explore what happens when you kind of give, if you, it's like being in the kitchen. If I
Dissect
give a little
mechanism.
stat one, a
Uh,
little stat three, maybe two scoops of stat four, what happens when I mix and match and what kind of T cells do I get out of that? And so we did a little bit of that in the paper where we took about, 20 different mutants and we found two that were really interesting because they had very similar stat three, four, and five, but they had very different stat one. And as Joe was describing it and trying to figure out what stat one does, that was the same thing we were going through. We're like, well, we kind of know what stat five does. We kind of know what stat three does. Stat one was a little bit more nebulous. We didn't really know why it was there and what it was doing. So we said, okay, well, now we have these two mutants, one with really high stat one with low stat one. And it turns out stat one plays this kind of like, it's like a dial. If you have too much of it, cells become really effector, like terminal effectors. They've done all their killing, they can't be useful anymore. Whereas if you turn it down too low, cells love to grow and expand and proliferate, but they don't turn into killers very
Right.
effectively. So you need a little bit of it to get cells to become effectors, but too much of it can be bad, and too little of can prevent them from being effective. So that was really informative. So it was a nice kind of peek under the hood of how this stuff works.
are there clinically available IL-9? Uh,
That's the biggest challenge. People have tried IL-2 in the clinic, IL-7,
Yes.
15,
15.
yeah,
21.
yeah.
All the gamma
Toxic.
chain cytokines have been in the clinic except IL-9. So there's no clinically available IL-9 that we can go use. So that's made it a little bit challenging to directly translate exactly what we report in the paper. Because if you imagine if you have to make a treatment, you have to make the cytokine, the protein, and you have to make the cell. And people who make cells are afraid of proteins, and people who make proteins are afraid of cells. So,
Well put.
so
Oil and water.
yeah, so it's a little bit tricky to get both of those, enough momentum to get those two parts, and then you need two FDA approvals. You need to test the safety of each, so it's a challenging process, but something that I really hope we can make happen at some point because I do think there's something special about doing it that way.
Uh, is one strategy to overcome that to make the CAR T cells produce IL-9 and have the receptor?
Yeah, and yeah, and that's a great question, and something we're actively kind of exploring is other ways to get the signal. My initial like impressions are that maybe it's very different. The biology is very different depending on how you give the signal, and
Huh.
so it works, but if you want it to work the best, you got to figure out exactly what the strategy is to deliver the signal.
So that would be the design, or how would you design? So say you had your dream, which is
Yeah.
the like engineered cells and your GMP grade cytokine ready to go, right?
Yeah.
So how long do you need to give it? Like, what do you think? Just a short course to get the expansion started? Do you think you have to give it
Not
continuously?
about expansion,
Uh,
Joe. Remember?
Yeah.
about persistence, but
Yeah.
for certain
Yeah. No,
indications.
if I could design a trial, I would do it a lot like we do it. I do think if you can give it basically through the point where the cells are in the tumor and trying to clear the tumor. And as soon as
Mm.
they've kind of started to clear the tumor, then you can stop giving it. But then you have it as a kind of a remote control. So
Right.
we found in our studies that you could wait 6 months later and give it to the mice, and the cells would just come roaring back into the blood when you gave the cytokine because they were all hiding in the tissues. You wouldn't see them in the blood unless you gave the cytokine, and then they came rushing back. So we think they're there. We think they could be effective still, but the I think you would just need to give it. And the nice thing about it is you have control over it. So, if a patient happens to have toxicity or anything like that, you can stop giving the cytokine. And, the cells will probably calm down immediately after that.
Do you think that that IL-9 would have differential effects for solid tumors versus heme malignancies? Do you think there's something
I
special
do.
about?
I do. I mean, I think there might be some advantages in the solid tumor space because IL-9 does seem to get cells into tissues really effectively, where I'm not a heme malignancy person, but I don't think that that is as major of an issue, although maybe bone marrow infiltration might be important in some cases. But for solid tumors, I think being able to get into diverse tissues and get to those metastatic sites is critical.
Wonderful. So, I think we've covered so much ground, but this is an immunologic tradition if you care to participate. So now that you've done all this beautiful work, what's the big question that keeps you up at night? So every time we solve one problem, obviously new questions emerge. And so is there a particular scientific or clinical mystery you most want to crack next? So is it understanding at the molecular level, which you've done a lot of already, why IL-9 is so effective? Are there any remaining mysteries in that signaling, or do you think you've cracked it?
Yeah,
Or is your focus really
yeah.
going to be on the practical deployment, like we alluded to,
There's
or both?
definitely way more mysteries than we can handle. I would say that probably there's two major questions that I've been thinking about a lot. One is we talked about it a little bit, which is how to deliver the cytokine.
Hmm.
But cytokines if you have like an infection, your T cells see cytokines for a brief period of time, and then they rest and they go away and they go do another job. Cells are not really used to seeing cytokines like constantly, um, like a fuel in that way. So understanding kind of the kinetics of the cytokine signaling and how that actually affects the fate, even if it's the same signal. For example, if it's IL-9, but it's a given in different patterns, what that does to a cell, I think is really important. Not just IL-9, but we have two trials recently, one from Penn and with the IL-18, where the CAR T cells are making their own IL-18, and then the one from Baylor from Andras Hex's group, who showed that get IL-15 secreted from the cells. So that's cells that are just pumping out their own cytokine and seeing their own cytokine for weeks at a time. Like what that's not normal. I mean, cells don't really you know, generally get exposed to the same cytokine for weeks. And we're talking about a signal that's like at the top of the food chain. It changes so much in the
Right.
cell. So it's not like editing one gene inside the cell, you're changing a whole program. So I think understanding what that looks like versus giving like pulses of cytokine,
Hmm.
temporally and understanding the differences in what happens to the T cells is really important. Um, the other thing that I am fascinated by is how now that we get rid of lymphodepletion, like we talked about at the beginning, a
Right.
human being who has a totally normal immune system or close to normal, and you're giving these souped up T cells, what is the interaction between the T cells and the host immune system now that you have that? I think there are opportunities either with CAR or even TCR engineered T cells to understand how they interact with the patient's own immune system in a beneficial way. So how can we leverage those interactions? We kind of always thought of these T cells as kind of soldiers that go to the tumor, take care of the job. They're kind of they do their own thing. But I think we probably underestimate the amount that the host immune system plays a role in shaping those cells. And especially if you can maintain an intact immune system, what we found is that the host immune system probably plays a bigger role than we previously thought. And so trying to figure out what those interactions are and how to leverage them is kind of something that we're thinking about a lot.
Hmm.
Can we go back to the trial? What would be the first indication that you would do this in?
That's a tough one. I mean, I think, um, yeah, that's challenging because I think, what car to pair it with if you were going to do it in a car, what TCR to pair it with,
Right.
heme malignancy versus a solid tumor. Um, do you pick something where cell therapies have been effective before? Do you pick a new target to test it out in? I mean, there's so many different avenues. I think for me, like I think I would, because I believe in the technology, I would want it to be something where I know cell therapy can work or has shown it could work, and we would want to
Mhm.
make it better. And
Sounds like
so I think
sarcoma
that's,
and a TCR is what you might be able
yeah, yeah. So as a sarcoma doc, that would be an
Right.
amazing trial. I would love to try it in the MAGE-A4 TCR.
Mhm.
Uh, the challenge there is the challenge we all know about, which is, even the companies that have been successful in that space are, and, have had a hard time sustaining their products because it's such a niche population. So, well, but I'm a big believer in doing things for rare diseases, and as somebody who takes care of sarcoma patients, I think that would be a fantastic space to do this kind of trial. We just need somebody or some organization to support that kind of work for a rare population.
It will happen. I know it will.
I was interested, I'm interested in hearing your perspective on, um, do you think there's anything unique about the sarcoma kind of immune microenvironment? I don't know much about sarcoma as primarily a lymphoma specialist, and all those like translocations and things scare me. So
Yeah.
teach us some more about sarcoma.
Yeah, no, sarcomas are, I mean, honestly, it's like 100 different cancers bundled up into one. So I think that is the biggest challenge with sarcoma is that each one of them presents its own challenges. But it's been really interesting to learn about the sarcomas that have responded to checkpoint blockade, for example, and the ones that don't, because these are mostly tumors that don't have high mutational burden, but there are some that bizarrely respond to PD-1 therapy, and we don't really understand why. And they don't have high mutational burden, they're not virally driven, they, don't have
A lot of tumors
microsatellite
including
instability.
lymphocytes.
They do. I mean, and sarcomas, there a subset of them do, but I mean, why they do, we don't really know. And is it because they're kind of these genomically unstable tumors? So I think that there's a subset of them that are these genomically unstable tumors that respond to PD-1 therapy, and then there's the fusion ones that you said scare you, which scare all of us because they are driven by these fusions, but we don't have any way of targeting the fusions. And then on top of that, the fusion ones tend to be immunologically colder. Um, and so that's a challenge. But I do think the fusion ones present a unique opportunity because I think they tend to be a little bit more homogeneous and so for cell therapy, I think that would be great. And I know Chris Klebanoff's group at Sloan Kettering has developed a TCR targeting one of the fusion oncogenes in one of the diseases that falls under the sarcoma umbrella, which is desmoplastic small round cell tumors. And I really hope that they can get that therapeutic into patients because it does look really promising. And I think that's perfect, right? That's a fusion oncogene as a driver, and so you could target it. And then there are several other sarcomas that have those. Finding the right TCR or CAR to target them is proven challenging. But I think we have some TCRs, so it would be great to pair some of those or one of those with the IL-9 system.
Absolutely.
The lymphoma is another one. It's a little tricky because you have therapies that work so well. It's like you've set the bar so high that
Yeah,
everybody's afraid to try their new thing in the lymphoma or the leukemia space. So that becomes a little bit
we
of a
are
challenge.
blessed with many wonderful, effective therapies, which cannot be similarly said for sarcoma. It's definitely true.
Yeah, that's true.
It's really, I'm so glad that there are folks working on improving our therapies for for diseases like, like all
Yeah.
these various sarcomas that are really challenging to treat.
Excellent. So any last minute thoughts?
I think
Did we
we've
not
covered a
cover
lot.
anything? Yeah, yeah. And I think we covered, everything from well, maybe not the nitty gritty of JAK-STAT signaling, but
Thankfully.
enough to understand. Yeah, thankfully, right? Different podcasts, right? But
Yeah.
we looked under the hood, as you put it, and also went into the very practical implications of patient care. And so, Anusha, thank you so much for sharing your expertise and really, more than that, your visionary ideas with us. And it's going to be
You're
exciting to see where you take this, right? And
And
when you take this to the
if we have
clinic.
any very, very wealthy listeners who want to support some groundbreaking and impactful research, you know who to contact to
Absolutely.
help
Well,
out.
you guys are too generous. It's a really honor to be here. You guys, and Joe's been somebody I've admired for a long time. So,
Oh,
thanks for having me, and thanks for
right
doing
back
this. I
at you.
think this is a great service also for the community. So,
Yeah, that's what we're trying to do. And to our listeners, all listeners, thank you for tuning into this episode of ImmunoLogic. As always, stay curious, keep pushing the boundaries. The field of immunotherapy, as we always say, is evolving rapidly, and what was experimental yesterday can be tomorrow's standard of care. So, Janna and I will be here to guide you through each discovery, one enlightening conversation at a time. And until next time, be well, stay immunological, and we'll catch you up on the next episode. Thank you and goodbye, everyone.